invitation to this webinar. It's really exciting. I'd like to thank Lukas because he gives such a great introduction to the topic. And this is exactly what we do as well. We want to address the problem at the root, like more, but we don't work in the field of cell growing and organized growing, but the handling, the cell handling. As soon as you have nice organoids, how can you handle them? How can you transport them from A and B in a very efficient and precise principle? Let me start at the beginning. We are a young company, as many others here too. Let us start by speaking about the people who really founded this company from the start, from scratch. And I hold a PhD in physics. I started off with a colleague who's also a physicist, and we thought we have these cool physical methods. We worked with traditional tools, so we really handled the tools, the instruments that are needed to handle cells as it was done at the time. And we then made a decision to look at organoids. And during the drug screening process, how can cells be handled in a better way? And 3D cell models are really crucial, particularly in the preclinical stage for pharmaceutical companies. It is there that they want to test what preparation is most effective. And we want to do this in an automated manner, but also scalable. We want to use the 3D cell models advantages. I'll get back to that later. But we want to stick to the costs of the 2D cell models. And that is something companies often struggle with, because you want to launch a new methodology, but you have to pass the status quo. You have to do away with the standard that already exists. And in order to do away with that initial barrier, you need technology. And this is what we do. We develop a device that makes it possible to make cell handling much easier, particularly the 3D cell cultures. And what we can do is several things. We don't only transport the cells from A to B, the cell models, we can classify them. Are these the models we actually want, the right type, the right properties? And we can then sort them and dose them. We do that for the mini spheroids. These are very simple cell models, very small. It depends, 100 to 300 cells compose one cell model. And we want to apply this standardized format. The 384 well plates is what we use, and we want to prepare the substance and work with it. And we prepare it for, prepare the drug screening or the preparation screening, and this always requires imaging. You have to make sure that you send the signals in different ways with different methods, but this is crucial. And then we started with the first applications. We are docked onto the pharmaceutical industry, and we use the ultra miniaturized liver spheroids. This is the tiniest substance possible. We've mentioned it already. We want to get away from animal models, not only for ethical reasons, but, and Lukas mentioned already, because it takes a long time, and the failing rate is really high. And in order to avoid this step, and in order to save time and money, the 3D models, cell models are increasingly popular. And here we see an opportunity, because spheroids are very positive. It's a much better model, not only in the academic field, it is viewed that way, but also in the industry. If you compare to traditional 2D models, you can imagine easily a 2D model is at one level, but in the human body, nothing is in 2D. You always have a three-dimensional structure.
Und das benötigt man eben dann, bessere Vorhersagen
And
treffen
this
zu
is
können. Also
what
diese
you
zwei
need
Themen,
in
die schon
order to make
angesprochen wurden,
better
einmal die
predictions, the two
Wirksamkeit
topics that
von
were mentioned already, the
Medikamenten,
efficacy of
aber eben auch
drugs,
die Toxizität.
but also their toxicity.
Und damit kann man ganz viele Assays dann
And many
gestalten.
different
Gehe ich jetzt gar nicht so im Detail darauf
assays
ein.
can be designed. That way,
Was
I'm
der
not going
Punkt
to
wirklich
dwell on
ist, dass
that
man
for too
dafür
much. But
aber
the
Werkzeuge
point is
braucht, ja,
that you
die
need the
eben
right
diese
instruments, the right
Variabilität
tools,
mit einbeziehen können
to,
von Patienten
which
und
include
dann
the
eventuell eben,
variability
wie das
of
schon gesagt wurde,
patients. And as has been said
Patientengewebe zu
already,
entnehmen und da Zellen zu
patient
entwickeln, um
tissue
dann wirklich
can be
spezifisch für Patienten
removed,
nah testen zu können. Ja?
and you
Das hat natürlich den Vorteil, dass
can
man
test it
dadurch
in
nicht
a
die typischen
patient-specific way.
Bias hat aufgrund
And
von Geschlecht
so you then
oder
don't have the typical bias like
Ethnizität etc.
gender,
Ja? Und
ethnicity,
das ist so
etc.
die große Vision
So
des
this
ganzen
is
Feldes eben
the
hin zu
big
personalisierter Medizin.
vision to move
Genau, wie
towards a
machen wir
more
das Ganze?
personalized
Also das
medicine.
sind natürlich
How
ganz
do we
viele
do this? There are
Herausforderungen, die
many
damit
different
kommen,
challenges that
weil so
come with
3D-Zellmodelle
this
erstmal schwieriger
because
sind
these
zu handhaben.
3D
Ja,
cell
man hat nicht
models
mehr
are more
diese
difficult to
typischen
handle. You don't have the
Petrischalen, sondern das
typical
ist schon
Petri
in einer anderen
dishes
Art und
anymore. You
Weise,
have
sodass
to
es
do
eben
it
auch
in a
eine
different way,
3D-Umgebung gibt, um die auch
so that there is
zu
the
kultivieren.
3D environment
Und das ist natürlich ein sehr,
to
sehr
grow it. And this
kostenintensiver
is very cost
Ansatz, den
intensive
natürlich
as
Moore
an
auch
approach,
schon sehr
and
gezeigt hat.
Moore already
Und man möchte natürlich auch
mentioned
reproduzierbar sein.
it,
Ja? Und
and
da
it shall be
wirklich das
reproducible.
3D-Zellmodell als ein
And the
genügsames Ersatzmodell
3D
auch anbieten kann.
cell model is something I want to
Und
offer
wir
as
sehen
a
das wie viele auch andere,
cost-effective
dass
replacement.
da ein gewisser Mangel an
And we
richtiger
see
Technologie
that
fehlt,
there
ja,
is a
oder da
lack of
ist.
technology in this
Und das
field.
ist eben beim Kultivieren, aber
And
eben wie bei
this
uns halt auch
happens
das, was wir
when
antreiben, ist das Bearbeiten und
cultivating,
das
but what
Präparieren von
we address
3D-Zelluntersuchungen.
is the
Ja?
preparation and the handling
Weil
of
eben
3D
dadurch
cells.
wertvolle primäre Materialien verschwendet werden
Because
können, Experimente
you
können komprimiert werden.
experiments can
Und
be
gerade wenn es jetzt um
done
diese
in a better
384
way, and
Wellplates geht,
particularly
die
if
man
when
zum
speaking about the
Beispiel diese Wells
384 well
verfehlt
plates,
oder eben mit mehreren besetzt oder die eben auch nicht
if you
lebensfähig sind,
have
dann hat man eben
more
an diesem Punkt keine Datenpunkt.
cells in
Ja? Das
one
heißt, man hat
well,
da natürlich
etc.,
viel
then you
Zeit,
don't
aber
have
auch
data
viel
point, and this
Kosten
is a waste.
verloren.
This is a waste
Und
of
natürlich,
time and money.
Automatisierung heißt auch immer, dass
And
man
also
menschliche
automation
Fehlerversuch zu
always
minimieren.
means that you
Ja?
try
Und
to
genau in
eliminate
so einer
human failure.
automatisierten und aber auch
And
standardisierten
in
Lösung
such an
arbeiten wir
automated
gerade.
and standardized solution, this is
Und
what
wie
we are
sieht unser
working on.
Modell eigentlich aus? Ja,
And
an der
what
rechten
does our
Seite
model
sieht man
look
so
like? On the
so ein
right-hand side, you see
3D-Rendering
the
auf so einer
3D
Box, weil
rendering
wir über
on a
Größe gesprochen
box.
haben. Uns ist es
We
ungefähr
spoke
so,
about a
so ein
size.
17 Zoll Laptop
This is a
als Fußabdruck
17-inch laptop
und so
as
ungefähr
a
50 Zentimeter
footprint and
hoch.
40
Ja, und das
centimeters
besteht
high,
aus drei
and it
Schlüsselkomponenten.
consists of three key
Das erste
components.
ist wirklich das Kernsystem, das ist sozusagen
The
das
core
Gerät an
system,
sich,
the
das ihr
device
rechts
as
sehen
such,
könnt.
is what you can see on the
Aber
right-hand
es hat
side.
eben auch ein ganz zentral ist eben
But it
diese
also
konsumierbaren Mikrofluidiken
has
Chips. Ja, das ist quasi die Umgebung, die
consumable
es braucht,
microfluidic
überhaupt
chips. This is
Flüssigkeiten und eben die
environment.
Zellen auf
It
kleinsten Skalen
needs
handhaben zu
to
können.
be
Wie
able to handle
natürlich
cells at
wirklich auf die
smallest scale.
Zellmodelle. Es gibt ja unterschiedliche
There are
Spheroide oder
different
Organoide und
spheroids
aber
and
auch unterschiedliche
organoids
Größen, die
in
damit
different
einhergehen. Und
sizes.
darauf passen wir das natürlich an.
And of course, we can
Ganz
adapt
besonders
our
ist hier,
device to the different
wie
needs.
viele andere vielleicht das auch schon
And
tun, ist
as many
wirklich KI,
others
also
might
künstliche
do as well, we also use
Intelligenzalgorithmen zu verwenden,
AI,
um dieses
we
ganze
use
Sortierverfahren
algorithms
und das Analyseverfahren zu
to
vereinfachen und damit auch
simplify
viel
our
schneller zu machen.
sorting and analyzing
Und somit kommen wir eigentlich schon zu diesen
process.
Schlüsselfunktionen, die wir
And this already
anbieten.
brings me
Das
to
ist
our key
erstens
functions.
das
That is
Qualifizieren von
the
wirklich
qualification
lebensfähigen Spheroiden.
of viable
Das ist ganz
spheroids.
wichtig, dass man quasi nicht
That is
nur
really
anhand der
important
Größe und der Form entscheiden kann,
to
sondern
not
quasi auch, was für
decide
Eigenschaften hat sie.
because of the shape or size,
Und
but also
diese dann
because of the
wirklich zu
properties.
sortieren, also von
And
gewünschten,
then
zwischen
to
ungewünschten und
sort
gewünschten
those
Zellen,
that we
aber
want to
eben auch
use and the
von
cells we don't want
einzelnen
to
Zellen
use,
oder
and
ja, am Ende
individual
Hybride oder
cells,
Dreck, der nun mal auch
or also the
normale
debris
Proben sein
or the
kann. Davon
dirt
müssen wir
that
natürlich
might be on
unterscheiden
the
können.
samples. And of course, we have to
Und
be
am
able to
Ende wollen wir die
differentiate.
natürlich wie quasi
And
ein
at the end of the
3D-Drucker
day, we
sozusagen
want to use it as a
einzelne
traditional
Tropfen
3D printer, individual
dispensieren. Das
drops.
heißt,
We want to
ein
dispense it.
Spheroid pro
This means
Well.
one
Und das
spheroid
ist recht
per
schwierig
well.
und daran setzen
And
wir
that
genau
is
an.
tricky and difficult, and this is
Und wie sieht
what
das
we
aus?
specialize in.
Diese
What does this look
Qualifizierung
like?
zum Beispiel ist hier, das ist so ein ganz
Qualification, for
einfacher
instance. Here
Mikrofluidic-Chip. Man hat quasi
you
so
see
eine
a simple
Kreuzung. Die
microfluidic
kommen alle von
chip.
links, gehen nach
It's a kind
rechts
of a
durch. Und wenn
crossroads.
man eben
They
dieses
come
Spheroid
from the
haben
left and
möchte,
go to
dann
the
schießt man
right. And
quasi
if you want
Flüssigkeit
to
von
have the
unten nach oben
spheroid,
durch
you
und kann die
inject a
damit
liquid
isolieren.
from the bottom to the
Was
top
eben
and
hier wichtig ist, dass wir eben
isolate
diese künstlichen
the spheroid. And we have artificial intelligence algorithms we use to classify the spheroids.
Ganz einfacher Basis gesagt, zum Beispiel nur die Größe.
For instance, just by the size. And depending on the position, we can decide if it is a spheroid we want to use or not. And in future, we also want to be able to make decisions in a label-free manner about the viability.
Spheroide, ich habe es schon angesprochen, wir können auch ganz andere, so sieht das jetzt
it's not only about spheroids. This is a live presentation. If you focus, zoom in and out, you see what the algorithm does. The algorithm tells us if this spheroid is close to the perfect state. This is just dirt on the sample, be it PTPMS or other particles. Of course, we can classify them too and make sure that they will be isolated from the system.
Das Ganze muss dann natürlich noch weiter sortiert und dann
we need to keep sorting and apply it on the well plates. We have two different systems that sort. On the right hand side you see the robot going through and applying drops on the well plate. This is a glass plate to show you the principle. This works very well. And we can also do it with the spheroids. We also have tumor spheroids we also used in real time. You don't see the isolation, it happens so fast. Here you see the whole system. This is the prototype. And it happens so fast that you cannot see it with the human eye. And here you see it from a different angle, from the side.
Hier können wir quasi wirklich ganz gezielt diese Tropfen auf die Probe bringen.
We can apply the drops in a very precise manner. And at this point, we are at 94% accuracy. And the process takes 2 minutes. So that's really good. That was a 96 well plate, not 384, but we are quite well underway regarding the time.
Okay,
so it's always nice to develop a technology, but it's also nice to see if that suits the customer. Automation is a big topic and a big criterion in the industry, because it's not only about the 384 plates, but also imaging is important. Others do the growing of the spheroids, others do that, and we qualify and sort them. That is a very important step in order to be able to prepare the 384 well plates and to save money regarding and time regarding imaging. We cannot only guarantee that it's one spheroid per well, but we can also guarantee the position. And then we have the imaging with a lower resolution. This is something you don't need to search for the spheroid anymore, where it is located, because we know exactly where it is located. So these are the key benefits. We are absolutely certain that the advantages of 3D prevail, and that we can stick to the costs of 2D. And the XYZ coordinates are very important, and we know the coordinates of each spheroid. And it stays the same even if you have different robots or automation technologies, if you move back and forth, and you can get relevant data of toxicity, drug screening, etc. And this already brings me to the end, and I'm really excited to hear your questions. And I'm also available for the breakout sessions, of course.
Ja, ganz herzlichen Dank.
Thank you so much.
An Falk.
Falk?
Ja, und wir haben auch ein paar Fragen, und zwar
Falco, and we do have some questions.
Und eine Frage ist,
and one question is,
man mit Physik oder Engineering,
let's say, how much knowledge do you have regarding physics and engineering to use these platforms? That's a good question. When speaking about automation, as Lukas has said already, you need a lab engineer, and you don't necessarily need to have a physicist having a PhD in physics. At the end of the day, it's a fully automated system. You have a reservoir where the spheroids are located in the suspension, and through the system and with the algorithm, thanks to the microfluidic chip, it is applied onto the well plates and placed. So what you need to do is to take the well plate, put it under the microscope, and analyze whatever you want to analyze. So then there would be another question. Let's say, if as a run-of-the-mill biologist, to put it that way, we could work with that without having to train a lot. Yes, exactly. It's supposed to be automated, and we are currently working on a simple graphical user interface so that you just need to click on the different models, for instance, and what do you want? Sorting is very important. What do you want to have? Do you want to have spheroids of a certain kind, mixtures of a certain kind, a certain cell size? And if you want to have a certain viability rate, that is also one factor, and you can just click and decide what you want. And this is quite a simple system, actually.
Dann noch, wie seid ihr zu den 3D-Zellkulturen
The
gekommen? Also wie
3D
sind
cell
da bei dir
cultures,
angefangen
how
und
did you
ja, wie kam
get to those?
das,
Did you
wo ihr
start
dahin
with
gekommen seid?
2D? What was the development? That is a good question. We didn't have anything to do with 3D cell cultures at the start. A colleague had worked with yeast cells, and then they looked into the microfluidic technology. And microfluidics, that is a big potential. Also regarding the big applications, where is it used? And then we thought about the pharmaceutical industry fast. Maybe you don't know that in Gothenburg, where our company is located, AstraZeneca is there too. Maybe you're familiar with that because of COVID. It's a big pharmaceutical company. And we do have good contacts to them, and we became aware quite fast that they have these problems. They have the spheroids, they cultivate them in droplets, but they didn't know how to handle them and how to apply them on the well plates. And we said, this is what we can do. This is our expertise. And then we specialized on that. Yeah, I mean, that's quite obvious if they're next door and they have the need. Then there's another question. What collaborations do you have in this field? Do you have collaborations with certain pharmaceutical companies, or do you specialize in certain areas? Well, we are involved in national collaborations with AstraZeneca as one of the big companies, the big players, but we also work with other research institutes in Stockholm, all this in Sweden. But for almost 3 years, we have been in a cooperation. It's called nanoscale drug testing. This is the cooperation about, and this is something that we want to enable. And it's about what we've just been presenting. It's about the work process, and we want to have an integration of the different work steps. That is our goal. We also work with other smaller startups in technological matters, the 3D cell models. How can we have them in one position so that they don't migrate or diffuse to the left or to the right? That's a very important aspect. And we will cooperate with others in order to secure this positioning. There's one quick question. Do you offer that as a service, or do you sell the devices? What is your priority?
nicht hundertprozentig raus. Also wir wollen, das kommt so ein bisschen auf
We
die User Case
haven't
an,
decided
ja.
yet. It depends on the
Wir sind jetzt noch,
user
dadurch, dass man
case.
eben ein Startup ist, man noch sehr flexibel. Am Anfang
As a
die Idee ist
startup,
schon, ein
you're
Gerät
still
zu
very
verkaufen,
flexible,
weil es
but our
einfach
idea is to
einfacher
sell the
ist. Ja,
device
also
because
man
that's
bietet etwas an, das automatisiert
easier.
ist, das
You
funktioniert
offer something that
und
has been
eben damit auch
automated, it
weniger
works well,
Wartung
and you need
benötigt.
less
Aber wir können auch
maintenance,
anbieten
but
Dienstleistungen.
we can also offer
Gerade wenn es darum geht,
services.
man möchte das nicht in diese 384
Particularly if you
Wellplatten-Standard
don't
haben, sondern
want
man
to
braucht
have the
etwas ganz
384
Spezielles.
well plates
Wir
as a
haben auch
standard, but you
viel
need something special.
Know-how, was diese
We do have a lot of
Microfluidic-Chips
technical
angeht,
knowledge
was die
regarding
Produktion
the
von solchen
microfluidic
Dingern,
chips and the production of the
diese
same,
Architektur quasi
the
und die Designs
architecture that
angeht.
is needed
Da können
regarding the
wir auch
design.
super helfen.
We can also provide support there, very well so.